Hello everyone. What we're going to discuss in this screencast is how do cells duplicate. So what we're going to be talking about is what is referred to as the cell cycle. We'll talk about the cell cycle first of all in prokaryotic organism, but then we'll spend most of the time talking about the cell cycle of eukaryotic organisms, which is a little more complicated, involves different phases. We'll focus particularly on the mitotic phase, which is that phase where the chromosomes are separated in eukaryotic organisms. And we'll talk about the structure that is responsible for this separation during mitosis, which is referred to as the mitotic spindle. So when we're talking about the cell cycle in general, in the end, what we're talking about is that really that amazing system that basically connects all living organisms. Is the system that basically leads from one cell to be duplicated into two cells, as you can see here. Imagine these cells being producing two identical cells here. And ultimately, this is really what connects every single organism on Earth to the original cell. Ourselves too. We're still connected to the original simple organism that was first generated about 4 billion years ago. Through this process, these organisms kept dividing, producing more and more cells, which along the way would have accumulated mutation, will be selected to produce more and more different type of organism. But ultimately, that's the same process that is connecting us and every single organism to that original cell. This is a very complex process, okay? We'll describe it as simplified, but it's a very complex process which in all organisms ultimately consists of three main steps. First, the cells, the original cells that has to duplicate will have to undergo some growth. You don't want a cell, obviously if a cell has to produce two cells, will have to duplicate not only the DNA but all its content. Because if it doesn't duplicate the organelles, doesn't duplicate the proteins, doesn't duplicate all the structure that are needed for another cells, this cell will get smaller and smaller over time, which is not compatible with life. Second phase will be the one where the DNA is replicated. And then, obviously, you need two copies of DNA to be delivered, identical copies to be delivered in the two daughter cells. And the last thing is how these cells get divided into two identical cells, two separate identical cells. So when we look at prokaryotic organism, these process is very simple. They undergo what is called a sexual replication by binary fission. So there is no mixing of DNA from two different parents. It's one cell that basically all it does, one bacterium, grows longer and then duplicates the DNA and gets separated into two. There is no mixing of DNA between different bacteria. So how is the cell cycle of prokaryotic? It's fairly simple. It consists of 4 different phases referred to as periods. So there is a first period, which is the one in which basically the bacteria will keep growing, elongating, getting longer basically in length. This is referred to as the A period. So is duplicating the proteins, all the necessary ribosomes, everything that is necessary to form two different cells. And then the duplication of the DNA starts in the second period, referred to as the B period. Here you start having DNA synthesis done exactly as we discussed when we talked about DNA replication. So we'll start from a replication origin. There is only one in the circular chromosome of bacteria. And this will produce 2 replication forks, which will keep expanding in the 2 directions. Right? Now, while this is happening, there is also the third period referred to as period C that starts, which is the one basically where the 2 chromosomes that are produced here, they get segregated into the 2 different poles of the cells. They start being pulling. Once this is done, the last period or period D takes place, and that's where the separation of the bacteria takes place. The 2 chromosomes have already been pulled on the 2 ends. You start having the separation of the bacteria taking place here, which will give origin to 2 bacteria right here. Now, each of these bacteria, if these bacteria do not find any condition to stop, they will go back and do the same thing. That's the reason why this is referred to as a cycle, the cell cycle, because these consist of a cell producing two cells, which then will go back to the same point and do the same thing, producing more and more cell. This is a very simple process, and you can see how in reality this division into different periods is a little bit schematic, does not reflect really the timing. For example, when you look at period B and C, you see that actually these are actually overlapping greatly. So you have that the DNA, while it's replicated, now you have some protein complexes here which would bind to the original replication. The DNA is opening in this direction, and while it's still replicating, so the replication forks are proceeding in the two directions, the DNA is already pulled. The two original replications are already pulled to the different poles. So period B and C are actually replicating, are overlapping ultimately. So a very simple overall cell cycle here consisted of very few 4 phases ultimately, which lead to the production of 2 different prokaryote cells. When it comes to eukaryotes, there are more complications, however. So the first complication is that, first of all, the DNA is much bigger and consists of multiple chromosomes. For example, we have 23 pairs, so 46 chromosomes in total, that have to be duplicated and separated. These chromosomes put all together, they are more than 1,000 times bigger than a chromosomal, the prokaryote chromosome. So you can see there is much more DNA to be separated. Second thing is that our DNA, unlike prokaryotes, is contained inside the nucleus. So there is a nuclear membrane too that has to be dealt with. So it has to be somehow removed first and put back. And so for this reason, eukaryotic cells in their cell cycle have developed an extra phase, which is referred to as mitosis. Mitosis is that phase of the cell cycle which deals specifically with the duplication of the nucleus and the separation of the duplicated DNA, so all the chromosomes which are contained in the original cells, the parent cell. We'll go through the steps of mitosis. Now, as we said before, mitosis, the nuclear membrane you see will disappear, will have to disappear, and will reform at the end, forming 2 different nuclei, which will contain different, 2 identical sets, ultimately, of the chromosome. You will see that this is done through a mitotic spindle and so on. We'll talk about these structures. Now, important information before we discuss the different steps of the cell cycle in eukaryotes is some wording here about ploidy and the type of chromosome we're referring to. So when you talk about chromosome, you will hear all the time, we'll be talking about homologous chromosome or sister chromosome or sister chromatids. What is the difference there? Well, when we talk about homologous chromosome, what we are talking about here, it's a pair of chromosomes which basically have the same genes. So here, this banding here corresponds to chromosomal regions which would contain ultimately the same genes. For example, the genes for the color of your eyes, the gene for your hair type, and so on. Now, they have the same genes, but then they have slight different type of genes in there. So variation of those genes, which are referred to as alleles. But the genes are exactly the same location. So here, the gene for the color of the hair is right here in both chromosomes, but they have slight variance. This one would have, for example, darker hair. This would have lighter hair. These 2 different forms are referred to as alleles. What we're talking about is differences in a few nucleotides between one gene and the other one, ultimately. So, similar chromosomes, but not exactly identical. They would have the same length, same genes, but different forms in them. Now, when these 2 chromosomes, and one of them obviously will be coming from each parent that have gave birth to you. So you would have one of these chromosomes coming from your dad and one coming from your mom. Now, once these chromosomes are duplicated, now they give origin to 2 identical copies of the one coming from your dad and 2 identical copies of the one coming from your mom. So in this case now, these two copies would have the same genes and exactly also the same alleles because they're identical copy of each other. So these two forms of the chromosome are referred to as sister chromosomes or sister chromatids. Okay, so you have two sister chromatids here and two sister chromatids here. Now, this pair would we refer again as two homologous chromosomes because two similar chromosomes but not completely identical. Okay. So now, this also relates to the concept of ploidy. When we talk about ploidy, we're talking about the number of copies of chromosome that we have per cell. So most eukaryotic cells are diploid, meaning that they have two copies for the same chromosome. We are diploid. We have two copies for the same chromosome. So we have in total 46 chromosomes with two copies of basically 23 types of chromosomes. We would have 22 homologous chromosomes and then two sex chromosomes, what we refer to as X and Y. Altogether, they give origin to 46 chromosomes. So you would have two copies, for example, of chromosome 1, one coming from your dad, one coming from your mom. Two copies of chromosome 2, one coming from your dad, one coming from your mom. Because of this, we are referred to as diploid. We have two copies of the same chromosome. Now, once you end up having a single copy of them, you will talk about haploid type of cells or organism. So you will see when we talk about meiosis is different from mitosis. Meiosis gives origin to cells, which are basically the cells, your sperm cells, your egg cells, that have only one copy. They would have only one copy of this chromosome, changed slightly, but only one copy of this. So by having one, they're referred to as haploid cells. There are organisms that are actually also, like yeast fungi, they're actually haploid. They have only a single copy of chromosome. Now, how are these chromosomes? These chromosomes that we have, so all of these chromosomes we have here, during the cell cycle are separated by a phase called mitosis. So now you have to understand one thing. So after DNA is duplicated through the process of the DNA duplication that we talked about in the previous lectures, now this gives origin for each of the homologous chromosome here will be copied, right? Gives origin to two copies, identical copies to sister chromatids, right? For each chromosome that are part of the same pair. For example, this could be chromosome 1. You would have two copies of each of the components of the chromosome 1. Now, these copies, once they're formed, they don't really separate. They don't go separate ways, but they stay connected with each other. So the two sister chromatids, they do stay connected in a region referred to as the centromere. This corresponds to a very specific region, sequence of DNA in these chromosomes, which allows them to pair together with the help of different proteins that are part of the centromere. These proteins are referred to as coenzymes. Okay, so the two identical copies, the two sister chromatids, they stay connected with each other. This is essential for mitosis because it helps with the separation of these two copies of DNA. And these are the two sister chromatids are the one that then in one phase of mitosis, referred to as metaphase, are aligned here, right in the middle of the parent cell. Now, these are separated, you see, through mitosis. This is just an overview of mitosis. We'll go through the different steps. And these, so that they're separated through a process referred to as chromosome segregation, okay, where you have one of each sister chromatids ultimately moving in one direction, one in the other one. Okay, let's look at the phase of the cell cycle in eukaryotic cells. So in prokaryotic, there are 4 phases, period A, B, C, and D. In eukaryotic cells, there are 3 main phases. The first phase is referred to as interphase. This is the phase equivalent, if you want, to period A of prokaryotes, where the cell is getting ready to divide. Or period A and B, if you want. So in this case, the cells is duplicated and the DNA gets duplicated in a specific subpart of interphase. Once everything is ready and the DNA has been duplicated in interphase, it gets separated by mitosis. Mitosis consists of different subphases, which we'll describe later on. After the DNA has been separated and two nuclei have been originated inside the same parent cells, now the cell gets split into two through the process of cytokinases. Cytokinases, unlike what is shown in your book, is shown as overlapping with mitosis, is not part of mitosis. It's a separate phase that takes place after mitosis. Mitosis deals only with the separation of the chromosomes and the formation of two nuclei within the same cell. Cytokinases separate the two cells and therefore the two nuclei into two different cells. So let's start with interphase. So interphase is that phase where basically the cells get ready for the separation of the chromosomes and the formation of two cells. It consists of three subphases referred to as G1 phase. This is where the cell grows. It will be duplicating its ribosomes, its organelles, and so on. It's getting ready to form two cells. Now, after the G1 phase, which can last different time depending on the cell type, what you have is that you have an S phase. The S phase is actually dedicated only to the duplication of the DNA and the proteins associated with that, so like histones and so on. And this takes pretty much the same time in all cell types, about 10 to 12 hours. And then there is another phase called G2 phase, which lasts about 4 to 6 hours. Some cells, like cancer cells, do skip G2 phase, but usually cells do have it, where you have a little bit of further growth before mitosis takes place. So after interphase, after all of these three phases, the cell will have now all the content duplicated, including the DNA, and could move on into mitosis. Now again, the S phase is the one that is dedicated to the duplication of DNA and therefore the formation of the sister chromatids, which is what we'll talk about later on in mitosis as well. Now, not all cells do replicate. So if cells, obviously what we're talking about here is the cycle of cells that produce into cells by cell divisions. But some cells do stop, do not duplicate, do not need to duplicate. They stay there quiet. So when they do that, obviously they don't go through all these steps of producing more organelles and so on. What they do directly from the G1 phase, they exit the cell cycle getting into a different phase referred to as the G0 or zero growth phase, which is basically they become quiescent, they become calm, they don't replicate, they might still be metabolically active, okay, but they do not produce any other cells. Then from this phase, based on other signal like hormones, for example, could stimulate that. Some cells can go outside of the G0 phase back to the G1 phase and enter the cell cycle, start duplicating. Okay, so this is normal, for example, for cells in your liver, for example. If you take some cells in your liver, you separate them, start growing until a certain point you have too many cells, and just the contact between cells will stop their growth, will get them out of the cell cycle and bring them into G0 phase. So after the interphase, now the cells have all the content necessary to form two different cells. They have two sets of chromosomes produced, and they have all two sets of organelles, ribosomes, and so on. So the next phase is mitosis. That's where the DNA, the chromosome, gets separated into two nuclei, starting from a single nucleus. Now, these can be divided into different phases. Your book refers to 5 stages, so we'll follow that, which are prophase, prometaphase, metaphase, anaphase, and telophase. So in all of these phases, what I want you to focus on is 3 main things. What happens to the nuclear membrane? Okay, you will see that the nuclear membrane in these 3 phases first disappears and then reappears. So by doing that, you start from one single nuclear membrane here, which contains all the DNA, and then you end up having here, after telophase, 2 nuclear membranes, each one containing one set of chromosomes, which then we'll get later on into 2 different cells. First thing, so what happens to this nuclear membrane? Second thing I want you to focus on is what happens to the chromosome. Now the chromosome here at the very beginning are not visible at all, but they have to be made easy to be moved around. And therefore, first they will condense. It's like when you take an airplane, you want to go, you don't take your clothes and you carry them all in your hands. You pack them nicely, right, into a bag, right? Otherwise, it will be complicated to move them around. You will be losing underwear at the airport. Same thing here. The DNA has to be organized so that the division of the genetic content takes place in a very organized way. And therefore, the DNA, the chromosome here, will become very compact through this, and then they will relax again at the end here. Made easy transfer. So second thing you got to look at is what happened to the chromosome. And the third thing is what happened to that structure that allows for the separation of this chromosome, which is referred to as the mitotic spindle. It will be appearing here and get into two separate poles, and then will slowly separate the chromosomes, and then once the separation is done, it will disappear here. Okay, focus on these three things. What happens to the nuclear membrane, to the chromosomes, and the mitotic spindle. Okay, first phase, prophase. So the cells here, they just finished interphase, and so they're getting ready to separate the DNA. So the DNA here will start actually condensing. It goes from basically being all invisible or indistinguishable in the nucleus into forming into this condensed structure here, which is what we always refer to as chromosome when we look at them. We'll start condensing. Not only that, but you'll start also seeing, as you can see here, this is the mitotic spindle. Is that structure which then later on will be separating the sister chromatids here. Will start appearing. So will start appearing at one pole here, and you start seeing that the mitotic spindle, which consists of different microtubules, we'll talk about that later on, these structures start moving towards the two opposite poles. Their function is to separate the sister chromatids on the two poles of the parent cell. So you want to have one set of chromosome on this end, one on this other end. So these mitotic spindles start moving at the opposite end so that the chromosome later on can be pulled that way. Second thing, the membrane, the nuclear membrane may start disappearing, but it starts really fully disappearing through prometaphase. So the nuclear membrane now, at this point, in the second phase, prometaphase, will have disappeared. And now you have that the chromosomes here, the sister chromatids, you remember this is two identical copies of the same chromosome for each pair, connected through the centromere. Now the centromere also will start connecting with that structure, the mitotic spindle, which then will separate the two sister chromatids on the two opposite ends. So in prometaphase, what you have is the nuclear membrane has disappeared. The chromosome, which now are already nice and visible, you can see them here as rods, they connect with the mitotic spindle, which now has already reached the two opposite poles. Then, on the third phase, which is referred to, sorry, I forgot to tell you that now the sister chromatids, so these are the two identical copies of the same chromosome, they do connect to the microtubules, which are part of the mitotic spindle, through structure called kinetochores, okay, which connect to the centromeres of the two sister chromatids and then will be responsible for them being moved. After prometaphase, so third phase of mitosis is metaphase. So in this case, again, focusing on the nuclear membrane, it's already gone, it's not there anymore. Focusing on the mitotic spindle, it's already separated in the two poles, you can see them here in green. And now you have the chromosome, which are already connected to the mitotic spindle, but in metaphase specifically, they get organized into the middle part of the cell. So this will be the middle part of the cell. The cell is this red thing here. Organized all right into the middle here, into what is referred to, this line here is referred to the metaphase plate. Not only this, but now what you have is that each sister chromatids would be on one side, if you want, of the metaphase plate. So they're going to be ready, basically, each sister chromatids are going to be ready to pull one towards one pole, one's towards the other pole of the cell there. And that's what happens in the next phase, which is referred to as anaphase. By the way, so these are the chromosomes that normally you are used to seeing what we call a karyotype. Karyotype is, again, the full set of chromosomes found in one cell, which is often done, for example, to see if there is any gross changes in chromosome structure or number. For example, if you want to investigate if a fetus has trisomy 21, so Down syndrome, that's what you would do. You would separate all the chromosomes. So you look at the karyotype and see if you have only 2 copies for each chromosome there. So going back, so after metaphase, then you have anaphase. Now the chromosome that are connected to the mitotic spindle get separated. So each sister chromatid, identical copy, one goes on one side, one goes to the other side. So this is basically what is responsible for the separation of the 2 sets of genetic material which were originated in the parent cells to 2 opposite poles, which then later on will become 2 nuclei. So now you start having basically here one genetic set on this side and one genetic set on this side. So they get segregated, they pull towards the pole, and then once they reach the opposite pole, the sister chromatids, you reach what is called telophase. In telophase, basically what happens is the opposite of prophase and prometaphase. So what happens is that now the chromosomes have been separated and they're done. They can start decondensing, so they become less visible than they were before. Same thing, now the nuclear membrane start appearing around the 2 sets of chromosomes that have been separated. This will correspond to the 2 nuclei of the future 2 cells. Third thing, mitotic spindle is not necessary anymore, will start disappearing as well. So as a result, at the end of telophase, you have 2 nuclei with the 2 sets of chromosomes here being separated within the same cell. And now the cells here can actually be finally separated into 2 separate cells through the last phase of the cell cycle, which is cytokinases. Now, cytokinases basically separates again the cytoplasm of the single parent cell into 2 separate cells, with each cell having one of the 2 new nuclei and having basically one set of chromosomes identical to the parent cell. How is this done? This is done differently depending on whether you talk about animal cells or plant cells. So animal cells do not have a cell wall. So all they have to deal with is basically the separation of the cell membrane. And this is done through the formation of basically a contractile ring of microfilaments in the inside of the cell membrane here, right in the middle. So imagine you attach a string inside the cell membrane there, and then you pull it, making it shorter and shorter and shorter, or the ring gets smaller and smaller. Since it's connected to the cell membrane, this will be pulling the cell membrane, forming like a girdle there, and this ultimately would lead to the fusion of the cell membrane right in the middle here, and the formation of 2 separate independent cells. This is how it's done in animals. In plants, it's a little bit different because, as I said before, you have not only the cell membrane, but each cell also has a cell wall. The cell wall is very rigid. So a system like this would only pull the cell membrane but not the cell wall. It would not work. So how it's done in plants is done instead through the formation of vesicles right in the middle here. So these vesicles obviously will contain a cell membrane, which is what forms the vesicle, and inside there would be accumulating material, which is the material that is found in the cell wall. So these vesicles keep growing and growing, and they will start fusing at a certain point. They will start fusing also with the cell membrane of the plant cell. And by doing that, you will start having basically 2 separate cell membrane here with a cell wall in between here, which later on will be separated, giving origin to 2 separate independent cells. Last thing we're going to discuss here is the structure of the mitotic spindle. So in mitosis, the mitotic spindle plays a key role on separating the two sets of chromosomes, the two identical copies of set chromosome there, which will basically be delivered to the two separate cells. How is this done? This is done through structure referred to as microtubules. So microtubules are nothing else than polymers of this protein referred to as tubulin. Tubulin is formed by two subunits. These are structures that are normally found in cells. Okay, they're structured, they're important in the formation of what is called the cytoskeleton. The cytoskeleton is that skeleton that is found inside every cell, eukaryotic cell, which basically gives the cell its shape, allows the cell to move around, change its shape, allows to do phagocytosis, allows for organelles to move around, ribosome to move around. It's a semi-solid structure inside that basically gives the shape and allows for movement of material and the cell itself. Now, this is done, again, by microtubules, which are formed by tubulins, which can polymerize and depolymerize. What does it mean is that, imagine, so this being your microtubule, imagine that this end is anchored somewhere. What you have is that when microtubules are growing, you have that these tubulin are polymerizing, elongating, so basically causing the formation or the elongation of the microtubule in this direction. If the opposite happens, so the tubulin depolymerize, so they separate from each other, you would have that this microtubule will appear to be getting shorter and shorter over time. Okay. This is the base of the formation of the microtubules in the mitotic spindle through polymerization and depolymerization of this tubulin protein. The same thing happens in the cytoskeleton. So these microtubules are organized around anchoring structures, which are referred to as centrosomes. Now, you have one centrosome in each cell when the cell is not divided, but when the cell is divided, this centrosome ultimately gets duplicated. And you will have that one centrosome will move to one pole of the cell, one to the other one. Now, the microtubules get organized, basically anchored to the centrosome, and they start polymerizing in the opposite direction. So this makes them look like they're getting longer or shorter compared to this structure. Since this is the organizing center of microtubule, this is referred to also as the microtubule organizing center, right there. So these centrioles, as we said before, they get duplicated. The centrioles, which makes up the centrosomes here, so there are two centrioles here, they get duplicated when the DNA gets duplicated too. If you remember, S phase is part of interphase. The DNA gets duplicated there. At the same time, the centrioles and the centrosomes get duplicated. So now you start having that each cell has two of them. And in the first phase, if you remember, or first two phases, prophase and metaphase, what you have is that these two centrosomes here will be moving to the opposite end. In metaphase, they are already at the opposite end. By doing that, what you have is also that the microtubules that are starting from there, they start forming from there, and they will be connected to the opposite poles there. So these are the microtubules, as you can see them in metaphase, basically, or the chromosome attached to them ultimately. Now, not all eukaryotes have the same structure in terms of mitotic spindle. There are differences. For example, in angiosperms, you have multiple mitochondrial organizing center or microtubule organizing centers present in there. But the one we're talking about here refers to most of the cells, so particularly animal cells. So again, microtubules, they do start, sorry, I repeat the same thing over and over, but it helps you memorizing this. They start from centrosomes here. They are organized from here, so these are polymers or tubulin ultimately, and they do form connections with the chromosome here at the centromere through the kinetochore. Now, there are two types ultimately of microtubules formed starting from the centrosomes, and they're referred to as the kinetochore microtubules. These are the ones that actually do connect with the chromosomes. And then there is a second type, which are referred to as the polar microtubules, which do not connect with the chromosome, but they have the function basically to push these two centrosomes away from each other. This helps with the elongation of the cells and ultimately the separation of the chromosomes as well. When it comes to the kinetochores, microtubules, again, these are the microtubules that do connect with the chromosome. So here you would have the two sister chromatids, identical copies, with their centromere connected to the microtubule to the kinetochore. So they're connected with this structure referred to as the kinetochore. This is the microtubule. Here you would have the sister chromatids connected at their centromere to the kinetochore here. So how they move to the opposite pole, so one sister chromatid in this direction, one in the other one, is because actually the kinetochore that connects them here can actually walk through the microtubule. So since it walks, whatever is connected to this, so imagine one sister chromatid here, would actually be moving along this microtubule. When this happens, at the same time, you have that the space behind the chromosome and the kinetochore ultimately will start depolymerizing. So this gives a sense as if the microtubule here is actually pulling the sister chromatids this way, as if it was an elastic. But in reality, is the sister chromatid walking through the kinetochore over the microtubule. How was this demonstrated? Was by then this very simple but elegant experiment where some scientists, what they did, they look at cell undergoing mitosis. So imagine this is the mitotic spindle and this cell. So this will be where the centrosome is. These are the microtubule and these are the sister chromatids that are going to be pulled towards this pole right here. So now what you can do, you can irradiate with UV light one part of the microtubule here. By doing that, actually, this part of the microtubule will look under the microscope as lighter than the rest part that was not irradiated. That's referred to as bleaching. And they will stay with this color. Now, if the microtubules were actually pulling the chromosome image as an elastic, what you would expect to see is that this bleached region should be getting shorter and moving slowly towards the pole. But in reality, what scientists observe is that this region stays exactly the same in size and in location compared to the pole, suggesting that this means that the sister chromatids are actually walking through this and there is just depolarization or disassembly of the microtubules here behind the sister chromatid here. So the second type, so what we talked about here are referred to as the kinetochore microtubules. The second type are polar microtubules, which do not connect with the chromosomes, but they have an important function. They actually push against each other. By pushing against each other, they're pulling the two centrosomes away from each other. By doing that, they create quite a bit of space between the two ends of the cells. They're pulling the cells too and creating a cryo region here where the new nucleus will be formed in telophase at the end of mitosis and another region here. So their function is just to pull the two spindles away of each other. I'm going to stop here in this lecture. In a separate section, I'll be discussing the regulation of the cell cycle. So this is a very complex process, as you can see, which basically ultimately has to lead to the separation of the genetic content into two separate cells, which have to have the exact same number and type of chromosome present in there. But you can see how easily you could have, for example, if we go back here to this step right here, you could easily have the two sister chromatids end up in one direction. Instead of being separate, both of them would end. This obviously would generate two cells, two nuclei first, and two cells with different content of DNA. So this has to be checked by the cells. Not only that, but also the duplication of the DNA in the S phase can be subject to a lot of mistake, a lot of mutation. Cells have to be aware of that, and they do deal with that with very, very, very tight checking of different steps, different transition between different phases of the cell cycle, which are referred to as checkpoints. And again, this is going to be discussed in the next lecture.